hopefully to be recognized by our immune system. Well, how does that happen? Well, you certainly have certain antigen-presenting cells within tissues. We talked about, for instance, in the epidermis, you have Langerhans cells, which are dendritic cells. We said, in fact, in the dermis, you can find macrophages. In most connective tissue, you find macrophages. In fact, the name of the macrophage, the type, is oftentimes linked to the tissue. You know that, for instance, in the lungs, we call them dust cells. In the central nervous system, we call them microglia. Well, they have different names, these macrophages, depending upon what tissue they live in. In fact, there are two subsets of macrophages. Some spend their whole life in tissues. Those are called fixed macrophages. Some move in and out. Those are called wandering macrophages. So if bacteria, as they first invade our sterile tissue, if they're not recognized by those tissue macrophages or dendritic cells, ultimately those bacteria make their way to regional lymph nodes, where again we find macrophages that hopefully at that point will recognize the pathogen. When I use the term pathogen, what does that term mean? Disease-causing agent, right. Now, let's suppose this is a little bacteria. Oh, this would be a little bacillus. You know what I mean when I say a bacillus? Rod-shaped organism. Okay. Well, and let's say it's being carried to the lymph node. Okay, so it's traveling through those sinus channels, subcapsular sinus, the trabecular sinus, medullary sinus. And remember, those sinuses are crisscrossed by reticular fibers upon which sit these guys. They're just on the lookout. They're performing surveillance. That's all they do. That's their job. That's how they roll. Okay? So if a bacteria makes its way past the macrophage, it's going to do this. What do you think it's doing?
Engulfing.
Yeah, it's eating it all up. Okay. And so it's going to be brought into the cell. Surrounded by a little vesicle. We could call this, in fact, a phagosome. And then they'll usually fuse with a lysosome. That's one of those little membrane-lined vesicles that have enzymes in it. And so when the two fuse, they form a phagolysosome. Okay. And within that fused vesicle, you can have a breakdown of the bacteria. It's pieces, little antigenic fragments. And then what this antigen-presenting cell will do, and remember, antigen-presenting cells have what are called MHC2 receptors, MHC2 receptors on their surface. Okay, they kind of look like this. Well, it will take one of these antigenic fragments and post it on its surface, linked to an MHC2. Okay. And in this way, it's going to present an antigenic fragment of that bacterial organism, of that pathogen, to the immune system. Now, when the immune system is first activated, what has to happen is an antigen-presenting cell has to present an antigenic fragment to what are called helper T cells. Okay? That's what's going to happen. It's going to present, and that will initiate the activation process. That will initiate activation of specific immunity. All right, so here we go. Okay? This is that antigen-presenting cell. Here's the antigen-presenting cell. Here's its MHC2 receptor. And look here. That little energetic fragment. Look good. Now, the helper T cell is also called a CD4 cell. CD proteins are different proteins that are present on various white blood cells, specifically lymphocytes. And so the type of CD protein present on helper T cells is CD4 proteins. And so sometimes we'll call helper T cells CD4 cells. Okay? And these CD4 receptors have a binding site for MHC2. So it allows the antigen-presenting cell to dock with the T cell. Now, the T cell also has a T cell receptor that will link up to an antigen bound to MHC2. So it's doing what I call the other day the double clutch. Okay, it's doing that double clutch handshake. And this will, and we'll talk about how this happens, but this is going to initiate specific immunity. This is going to trigger specific immunity, this double clutch here. Now, let's go back. And with regard to specific immunity, there are two major types of T cells. Now, for T cell, the CD4 T cell is the one we just looked at. Okay, their job is to recognize antigenic fragments associated with the MHC2 receptors on the surface of antigen-presenting cells. Okay? Now, when that helper T cell becomes activated, okay, as a result of that double clutch, it begins releasing a variety of cytokines. A cytokine is an inflammatory mediator. It helps to generate an immune response. Okay? Now, sometimes these cytokines are called interleukins. This isn't showing up well. Traditionally, an interleukin was a cytokine released from one white blood cell that targeted another white blood cell. And so that's where the name is derived, interleukin. So an interleukin is simply a type of cytokine that can be released by a white blood cell, like a lymphocyte, okay, and have an impact upon another type of white blood cell, another type of lymphocyte. So when the T cell becomes excited, it secretes a variety of cytokines, including interleukins, that are going to enhance activation of the cytotoxic T cells, the ones that perform a direct attack, as well as B cells, the ones that differentiate into plasma cells and produce antibodies. Everybody follow all that? That was a lot of stuff. Now, we said that helper T cells are also called CD4 cells. The other major type of T lymphocyte is a cytotoxic T cell, which has a different type of CD protein on its surface. It's called a CD8 T cell. These are also programmed to recognize foreign antigens, but not on the surface of MHC2 receptors. They're trained to recognize foreign antigens associated with MHC1 molecules. Okay? And these remember MHC1 proteins are located on all nucleated cells. Specifically, cytotoxic T cells are going to recognize antigens linked to MHC1 molecules on body cells infected by microbes. Remember we said they're involved in intracellular infections. So in most viral infections, because viruses have to live inside of living host cells, part of that response, that immune response, is a cytotoxic response, is a cell-mediated immune response carried out by cytotoxic T cells. They also recognize abnormal antigens linked to MHC1s on tumor cells. And then they also recognize these same on cells of transplanted tissue. Now, problem with transplanted tissue is it's going to have the wrong, in this case it's MHC2, but for a cytotoxic T cell, that transplanted tissue is going to have the wrong MHC1. Okay? And so a cytotoxic T cell is going to recognize it immediately. Now, whether we're talking about a cytotoxic immune response, an immune response carried out by cytotoxic T cells, or a humoral immune response that's carried out by B cells. Okay? In order to generate this type of immune response, two things are required. The cytotoxic T cell or the B cell have to come into contact with the antigen. Okay? Well, we said cytotoxic T cells come into contact through MHC1 receptors. Okay? A second thing has to happen, that they require co-stimulation by interleukin-2 produced by helper T cells. So helper T cells have to also come into contact with that same antigen, and when doing so, they release interleukins that then co-stimulate either T cells or B cells so that they can become fully activated. So it's not only that for the cytotoxic T cells and B cells that they have to be acquainted with the antigen, they have to come into contact, but then they have to receive stimulation from a helper. Okay? It's like you're going to be assigned a job. Let's say to move all these chairs into the next room. Okay? And, okay, someone gives you a set of instructions, you know what to do, but it's just an overwhelming task for you. And someone else comes, a little interleukin says, I can help you. And all of a sudden you're moving the chairs. You're good. You're fully stimulated. You're excited. Okay? By yourself, you have nothing. but with that co-stimulation, you're good. All right? All right. So, you know, they say a picture is worth a billion words, right? Okay. So what we're seeing here is that B cells and pre-T cells are being born in the bone marrow. The B cells mature there, whereas the pre-T cells have to travel to the thymus, okay, to become fully immunocompetent, to mature. And then these cells then travel to secondary lymphatic tissue, like lymph nodes and spleen, where they carry out immune surveillance. Okay. Now, this is a CD8 cell. So what type of T cell is this?
Cytotoxic.
Cytotoxic T cell. So if in that role, performing the surveillance, it comes into contact with a pathogen, with a foreign antigen, it's going to react. It's going to generate a response. But first, look, it needs help. It has to receive co-stimulation in the form of interleukins from a helper T cell that's also recognized that antigen. Now, once that happens, once it receives co-stimulation, then that T cell is activated and it begins to proliferate and differentiate into the cytotoxic T cell that performs the attack. Fully activated. The same thing is true with B cells. Okay? B cells are going to recognize an antigen. They're going to come into contact with an antigen. They have receptors on the surface that recognize an antigen. And then because these are antigen-presenting cells, they can present that antigen to a helper T. Okay, that helper T then releases interleukins that activate the B cells. The B cells proliferate and differentiate into plasma cells. Okay, those plasma cells begin producing these little Y-shaped molecules called immunoglobulins. This is part of humoral immunity. Those immunoglobulins will then seek out and coat the antigen wherever it's found. This slide's a little bit complicated. Okay? It's always a little scary when you see a real complicated slide with lots of stuff going on. It can be overwhelming. And so the key with a complicated slide is just to take it one little piece at a time to make ourselves feel a little bit more comfortable. So we're going to ignore most of the slides.
So we're going to go to the club.
Huh?
So we're going to go to the club with an example.
No. We're just going to focus here on this little bitty part right here. This is an antigen-presenting cell, like a macrophage, that has just processed an antigen. okay? And it's placed the antigenic fragment on the surface in conjunction with this MHC2. Look what it's doing, it's now presenting it to the helper T cell. It's now presenting it to the helper T cell. Okay. Now, look what's happening. As a result of this double clutch, that antigen-presenting cell releases interleukins and other cytokines, that stands for interferon, that then activate the helper T. Okay. And then, once activated, the helper T then divides and differentiates, perhaps along different lines. Notice that some of these daughter cells are what are called helper T cells, helper T1s. You have even memory cells. Okay. And the purpose of these cells, these helper T cells that are being generated as a result of this contact, is to release cytokines. Okay. We'll come back to that. At the bottom of this little diagram, I'm showing you this could be a macrophage, or it could be an infected cell, okay, any type of cell that has been exposed to a pathogen that's inside. And it's presenting a fragment of that on its surface in conjunction with an MHC1. Okay? Who's recognizing it? This is a cytotoxic T cell. It recognizes antigen fragments on the surface of MHC1s. So it's coming into contact with a potential pathogen right here, but it's not fully activated. Okay? To become fully activated, it has to receive interleukin 2, which is released from one of these helper Ts. Now, look at this. You see how it's got all these little fringes all around? It's kind of getting bigger and getting stronger and dividing and differentiating. Okay? To form a fully activated T cell. Now, that fully activated T cell is going to search out other cells that have that same antigenic fragment on its surface. Other cells that are similarly infected. Okay? It's going to bind